Good evening. So tonight, the Bible and Science, Lesson 4, Organic or Chemical Evolution. This is a supposed evolution of life. This is what pastors refer to as abiogenesis. So that's where we're going tonight. That's the map. Lesson overview. So last week we looked at stellar and planetary evolution. This week, organic or chemical evolution. Next week, macroevolution, or the supposed changing of one kind of animal to another, or the changing in phylum. Then week 6, we'll look at scientific laws in the Big Bang. So we'll revisit the Big Bang just one more time to really show you the scientific laws that the Big Bang violates. Then we kind of go in a different direction on lesson 7, and that's 6 literal 24-hour days, question mark. And so we'll be addressing what does God say about the creation and how long He took to create. Working definition. So you've seen the definition of science. We'll do that again tonight. You've seen the definition of evolution as it's continued to develop. We'll look at that tonight. 2 new words, abiogenesis. We'll look at Webster's Dictionary for that. And natural selection. We'll look at that as well. So science, again from Webster's. Knowledge about the natural world that is based on facts, learned through experiments and observations. And so you'll see that I continue to ask, as we look at these things, is this science? Does this fit the definition of science? The key words there, facts, learned through experiments, and observation. Abiogenesis, a little bit longer definition, but you'll get it. The origin of life from non-living matter. Specifically, a theory, that's interesting, Webster's recognizes it immediately, in the evolution of early life on Earth. Organic molecules and subsequent simple life forms first originated from inorganic substances. Water, rock, electricity. Those are the substances in play. Natural selection. Survival of the fittest, maybe you've heard it described as. A natural process that results in survival and reproductive success of individuals or groups best adjusted to their environment, and that leads to the perpetuation of genetic qualities best suited for that or to that particular environment. Now, we would not disagree with that definition of natural selection. We would agree that long-haired dogs will do much better and they're much more likely to survive, and you're much more likely to find nothing but long-haired dogs in the end in a super, very cold environment. We would recognize that in a hot environment, over time, all you would have is very short-haired dogs because the long-haired dogs did not do well. They died out. That information was not passed on. So we don't have a problem with this definition of natural selection. I do want to be clear on one thing, and that's that you understand what's required here. It's an observable process, and look at it. Survival and reproductive success. So we're talking about an organism, even if it's a cell, that can reproduce before we can ever talk about natural selection. So you can't talk about natural selection as how life came about, because there was nothing that was reproducing at that point. Natural selection only comes into play after you have life. And so you'll often hear them throw out, well, it's natural selection. No, not at the genesis of life. That's not possible. That doesn't fit your definition of natural selection or survival of the fittest. So evolution, we looked at the Big Bang, the origin of space-time matter, cosmic evolution. We looked at stellar and planetary evolution. And then tonight, chemical evolution, abiogenesis, the origin of life. And that's where we find ourselves tonight. This next video clip is from Discovery Channel. So in this short video clip, you have really one of the many explanations, and they're all kind of similar, of how the first living cell came into existence.
"4 billion years ago, a ball of rock and dust spins in the frozen vacuum of space. This isn't Mars or Venus. This hell is Earth. This seething, lifeless mass of molten lava will become home to nearly 9 million living species. But generating life from this will take a chain of events that defies the laws of probability. There are many theories. Nobody knows exactly how or where life began. But it couldn't happen without water. Experts believe asteroids or comets delivered it here. The water is churning with chemicals and organic compounds. Lightning strikes the chemical soup at the right place and the right time. Billions of volts of electricity trigger a chain of improbable coincidences. The chemicals' atoms join up in a precise sequence, creating a bundle of genetic material. These fragile genes don't stand a chance in this extreme environment. But luck strikes again. A blob of oily material engulfs a single chain to create the first ever cell. Now, the genes send out messages, chemical instructions. And 3.5 billion years ago, they do something extraordinary. They copy themselves and the cell to create a perfect clone. This is the very first living thing. Every human, every animal, every bug, every plant can trace its origins to this single cell.
Wow. Okay, so let's review. And again, I'm only going to quote the video. Generating life from this will take a sequence of events that defies the laws of probability. So let me sort of, I guess, rephrase that for you. That is statistically impossible. Lightning strikes the chemical soup at the right place in the right time. Billions of volts of electricity trigger a chain of improbable coincidences. Statistically impossible likelihoods. The chemical atoms join up in a precise sequence, creating a bundle of genetic material. Interesting. So what are they talking about? Well, genetic material, they're talking about the genome of this first cell. It's important for you to understand that the most basic cell has approximately 5,000 coded letters that make up its DNA. 5,000 coded letters that must be precisely coded to have life in that mitoplasm, in that simple cell, the simplest life form we know of. Now, if you think about 500,000 letters, and I think I said 5,000, so please forgive me, 500,000 letters, on a typical page of paper, single-spaced and typed at 12-point font, you have about 3,000 letters. So picture a 166-page book, single-spaced, that came about all the letters by chance. That's the statistical probability we're talking about here. To create this first cell and all the genetic information that was required at this moment in time. And we're not even talking about the DNA molecule itself yet. We're not even talking about the RNA molecule needed to copy the DNA. We're just talking about the genetic information itself. A 166-page book for this, and it had to come about by random chances, and it had to be right the first time, no letters wrong. These fragile genes don't stand a chance in this environment. Yeah, because you've got this gene, this pile of genes, this DNA, with no cell membrane around it. It stands no chance. Let me help you. But luck strikes again, and a blob of oily material engulfs this single chain to create the first ever cell. So luckily enough, this blob of material that engulfs this DNA does not destroy it, but in fact harbors it and creates a case for it that will be used later and even reproduce some of this oily material as cell division occurs. The genetic material, and you'll see this tonight. The complexity we're talking about that is going to be required to now live outside the cell for this period of time makes this an absolute, as I said here, pipe dream. Now the genes send out messages, chemical instructions, and 3.5 billion years ago they do something extraordinary, impossible. They copy themselves and the cell to create the perfect clone. So what they've done is not only have they given you all the information, they've created the DNA molecule, they've created the RNA, and all the micro machines you're going to need to replicate this DNA in an instant. This is not possible. There's no way you will get here from this. This is the very first living thing. Every human, every animal, every bug, every plant can trace its origin to this cell. My thoughts. First, this is a fairy tale. This is not improbable coincidences and luck. Instead, this is an outline of events that are absolutely impossible. But there is good news here. This theory is testable. We could not create the cosmos in the lab. We can create the supposed conditions of the early Earth, as you saw it here, a molten ball of mass with comets bringing the water. We can recreate this in the lab. We can shock the water. We can attempt to simulate this event. This theory is indeed submittable to scientific examination. So here's the timeline. We've talked about this before, 13.7 billion years ago, the Big Bang, hydrogen and helium, 13.2 billion years ago, the first stars, but we've already seen, 13.2 billion years ago, the first stars formed, but again, we've already seen that already galaxies were forming by the Big Bang's timetable. So already this is all confused on itself by their timeline. Galaxies forming 11 to 8 billion years ago, the Milky Way somewhere between 9 to 7, the Earth forming 4.5, and there we are tonight with the first cell engulfed in this oily material 3.5 billion years ago. That's their timeline. This is Paul Davies. He's a scientist. He's an atheist. And here's what he says. Nobody knows how a mixture of lifeless chemicals spontaneously organize themselves into the first living cell. As you'll see his next quote, he's not a friend of Christians in any shape or form, but he recognizes there is no theory to get a cell, even a simple cell, through chance and random processes. A lengthy quote, but I'll read it. It's from The Fifth Miracle, The Search for the Origin and the Meaning of Life. Isn't that interesting for an atheist to entitle a book? The Search for the Origin and Meaning of Life. I happen to know where they can find the answer to that search. But let's get back on our study. The abiogenesis problem is now so serious that most evolutionists today tend to shun the entire field because they are, quote, uneasy about stating in public that the origin of life is a mystery, even though behind closed doors they freely admit that they are baffled. Because, quote, it opens the door to religious fundamentalists and their God of the gap pseudo-explanations, and they worry that a, quote, frank admission of ignorance will also undermine funding. That's Paul Davies. That's an evolutionist, no friend of Christians. You can see how he refers to Christians. That's what he says. No explanation, and they don't want to say so because they're afraid a Christian will go, I have the answer. Now, maybe you've heard about Stanley Miller's experiment, University of Chicago, 1953. Anybody heard about it? Origin of Life series, right? So let's go to it. So in 1953, Stanley Miller, who was a PhD student at the time, and Harold Urey, who was his PhD advisor, decided that they could replicate those early Earth conditions. Now again, we know what the early Earth looked like. The Bible's clear. It looked very similar to what we have now. We'll talk more about that in a later study. But what they believed it looked like was an atmosphere that was comprised of hydrogen, methane, and ammonia. This would have been an extremely hydrogen-rich environment where you and I could have not survived. And so they took those gases, ammonia, hydrogen, methane, and placed it in this sealed system. They also placed water in this lower beaker, drew a vacuum on the entire system. Next, they inserted this rod that would be used to boil the water. And so what they would do is generate steam, press that steam and all the gases through this spark chamber, and in that chamber simulate lightning with 60,000 volt discharges. And they did this for a week straight. At the end of the week, they opened it up and they found nothing. So they did some adjustments and did it again and again, but ultimately, after a full week of multiple attempts, they did find some molecules and a few amino acids. The two amino acids principally, though, were glycine and alanine. So that's what they did. That's what they found. So no complex biomolecules, not the 20 amino acids that are needed to create a complex biomolecule, no cells, no life of any kind. A few molecules and amino acids, principally two, glycine and alanine. So now, I'll give you Stanley Miller's quote later in life. This is the guy who earned his PhD on this experiment. It turns out the problem of the origin of life has turned out to be much more difficult than I and most other people envisioned. You see, he was certain he would be able to replicate life in the lab. 60 years later, we've still never been able to do anything more than what he did. This next video is from Evolution's Achilles Heels. You'll hear from Dr. Jonathan Sarfati and Dr. Robert Carter.
The general theory of evolution says that all living things came from a single cell, and that cell came from something like a primordial soup. And for me as a chemist, the hardest problem for evolution is the origin of first life from non-living chemicals, and this is commonly called chemical evolution.
Most people have heard of the Miller-Urey experiment, and it's in most textbooks still today. They wanted to see if organic molecules could come from inorganic precursors. So they took some simple chemicals and they ran them through a spark chamber. And most of the products they got really amounted to nothing more than a brown tar, because random chemical reactions produced random molecules. And yeah, they found a few amino acids, but in a mixture of left and right-handed forms. But what they didn't find are certain specific amino acids that are critical for life. They didn't find nucleotides. They didn't find any large biomolecules.
To get living things, we need lots of huge molecules, and that means we have to get little molecules to combine into big molecules. And the problem is that chemicals don't react in that way. Everything I've learned in real chemistry shows that the reactions go in the opposite way from what's required for life to come from non-living chemicals. Any chemist trying to do that would not have water in the reaction, because water tends to drive the reaction in the opposite direction towards the little molecules, and yet the primordial soup would inevitably have loads of water in it. So it's the last place any real chemist would try to make proteins or DNA. Another problem with chemical evolution is called the chirality problem. Now, amino acids in proteins and sugars in DNA come in two mirror image forms, like your left hand and right hand. But all the amino acids in our body are left-handed, and all the sugars in DNA are right-handed. But a primordial soup would have an equal number of both, and it's quite complicated to get a single-handed mixture. And a primordial soup does not have that know-how, and therefore it's totally unsuitable for the origin of life.
So again, a couple of good points there, but I do want you to see a couple other things before we wrap this all together. This video clip is from The Case for the Creator, put out by Lee Strobel. Here, this is Dr. Jonathan Wells talking about Miller's experiment in 1953.
Strobel's search began with an examination of evidence that challenged materialistic theories of life's origin. He discovered that this negative evidence contradicted the textbook explanations that had once convinced him the blind forces of evolution could account for the creation and diversity of life on Earth.
A good example of negative evidence is the 1953 Origin of Life experiment by Stanley Miller, the one that helped lead me into atheism in the first place. As biologist Jonathan Wells explained to me, Miller's experiment has now been thoroughly discredited.
Stanley Miller put together a glass apparatus, and in that apparatus he put a mixture of gases that people at the time thought reflected the atmosphere of the early Earth. And those gases were methane, ammonia, hydrogen, and water vapor. But then the professional opinion of what was there on the early Earth changed in the 60s, geochemists revised their hypothesis and decided that the hydrogen, being very light, would have escaped into outer space. The Earth's gravity isn't strong enough to hold it. And probably the early Earth's atmosphere then consisted of what we now see coming out of volcanoes today, namely carbon dioxide, nitrogen, and water vapor. Well, if the early Earth's atmosphere consisted of those gases, then Stanley Miller's experiment would not work. In fact, he himself tried it with those gases, and he found that he couldn't produce any amino acids at all. So the experiment falls apart once you use a more realistic mixture of gases in the apparatus.
Miller's test has been repeated many times using the correct atmospheric components. The results are always the same. The amino acids that generated so much enthusiasm in 1953 do not appear.
Even if Miller's experiment were valid, you're still light years away from making life. It comes down to this. No matter how many molecules you can produce with early Earth conditions, plausible conditions, you're still nowhere near producing a living cell. And here's how I know. If I take a sterile test tube and I put in a little bit of fluid with just the right salts, just the right balance of acidity and alkalinity, just the right temperature, the perfect solution for a living cell, and I put in it one living cell, this cell is alive. It has everything it needs for life. Now I take a sterile needle and I poke that cell, and all its stuff leaks out into this test tube. You have in this nice little test tube all the molecules you need for a living cell. Not just the pieces of the molecules, but the molecules themselves. And you cannot make a living cell out of them. You can't put Humpty Dumpty back together again. So what makes you think that a few amino acids dissolved in the ocean are going to give you a living cell? It's totally unrealistic.
Okay, so just one thing before we look at one more video clip and sort of tie this all together. So please keep in mind, is he's talking about, or I'm talking about, the early atmosphere. That would be the early atmosphere by the evolutionary theory, by their own theory. So once they shifted the atmosphere to match their revised theory, it no longer even produces amino acids. Now you won't read about that in the textbook and you won't find that on the internet. But again, that's their theory and that's their atmosphere, and when you bring those two together, now we've taken 2 steps backward, not 1 step forward. And we'll talk more if you actually put our atmosphere in there. So we'll talk about that in a moment. It is important to note, though, Lee said that this study, the Origins of Life study, was one of the principal things that led him to unbelief. Finally, this video, it starts with Dr. John Sanford, really some final comments on this experiment, and then we're going to sort of tie it all together.
It's not sufficient to invoke scenarios where I could imagine biochemicals arising spontaneously. Life isn't based upon biochemicals. You can have all the biochemicals you want, and it's not going to give you life. You can have all the amino acids you want. You can have all the proteins you want. You can add RNA to the soup. You can add DNA to the soup. You can even add membranes to the soup. But number one, they will never assemble into a coherent, correctly assembled cell. And even if they could, you still wouldn't be anywhere near creating life because you have not introduced into those molecules information.
Now think about information in the form of books, which is ink molecules written on paper. The information didn't come from the ink molecules. You could pour ink on a page and you would not get a book out of it because the information in the books is the result of a mind organizing those ink molecules into letters and words, sentences and paragraphs. For instance, the simplest living cell, the mycoplasma, has about 600 kilobytes of information.
It's like an alphabet. The letters do not say anything at all. It has to be in the right position and it has to be interpreted. And that is what is going on in the cells of living beings.
You see, there's nothing in chemistry alone that would put any sort of coded information into a molecule. I mean, even if you could get nucleotides to form in a chemical soup, and even if you could get them to form a chain, you'd have nothing but a random string of letters. If you were to wait for a very long time and by chance produce a coherent string with a real instruction, even then, it would be just one clear message in a sea of random messages. This is the opposite of what life requires. Life requires lots of information, tightly controlled and protected, able to be copied, able to be fixed when an error appears. And all of this must be present the first time life appears.
When the DNA was first decoded and the scientists started looking at the simplest living cell, they imagined that possibly we could have a cell with a couple of hundred proteins. Well, it's up to over 400 now. And even the origin of one of these proteins by some chance process, it's not going to happen even on a universal time scale. And even if every atom in the universe was an experiment for every possible molecular vibration of its supposed billions of years of time, you would never get one protein, let alone hundreds, let alone the DNA to actually code for them. I mean, the idea of the origin of life by natural processes is a preposterous idea, absolutely preposterous idea.
Okay. So in summary, Miller's experiment created nothing more than a few amino acids, principally glycine and alanine, and those in very small amounts. We now know without all 20 amino acids, you will not form the proteins required for life. And that's just the proteins. Miller's experiment created nothing more than a few amino acids in a mixture of left and right-handed forms called a race mate. All amino acids in our body are left-handed, all sugars are right-handed. A mixture of left and right-handed amino acids is the exact opposite of life. It's toxic to life. Miller's experiment used what evolutionary scientists believe were early Earth conditions. Evolutionists no longer believe those are the correct conditions. If you rerun the experiment with the conditions they now believe, you do not even create any, zero, none, no amino acids. Miller's experiment used what evolutionary scientists believe were early Earth conditions. Well, what if you actually run them with something close to our actual conditions today, with oxygen in the atmosphere? Well, guess what will happen when you shock that oxygen in that cylinder? Not only will you not create amino acids, you will blow up Stanley Miller's apparatus. Thank you, brother. Miller's experiment created a few amino acids. This is a long way from a single complex protein, which requires all 20 amino acids, and a long way from a living cell, the simplest of which we know about, contains 400 complex proteins. By the way, a typical human cell contains 100,000 proteins. And we have not created a single protein yet. Even if you could create all 20 amino acids required for organic life in left-handed form only through random processes, which you can't. And even if you could, from those amino acids, create the hundreds of complex molecules needed for organic life, which you will not. And even if you could create a membrane that would wrap itself around this thing all at one time to encapsulate those molecules, which is not going to happen. And even if within those molecules you happen to have a DNA molecule and you happen to have the RNA to replicate the DNA, which, let's be honest, who are we kidding here? You are still missing one vital thing, and that is the information contained in the DNA cell or DNA of every one of us. The human DNA has 3, take a look at the letter, billion letters. So somebody help me with the math here. I told you that the average sheet of page, single-spaced, had 3,000 letters on it. If I were to write your DNA out tonight, how many pages would that book be? How about 1 million? Drop the 3 zeros, drop the 3 to a 1, 1 million. A 1,000,000-page book by random processes. That is serious good luck. But let's dive a little bit further into this information conundrum. Again, this is from the video Evolution's Achilles Heels. Now let me say something before we look at this. We're going to talk about information one more time at the very end, and we're going to spend a full hour really looking at the complexity of information within the genome. But again, right now, just to brush you over some of the facts.
Many evolutionists claim that the origin of life is not part of evolution, but come on, they believe that all living things came from a single cell, which in turn came from a primordial soup. So they have to have a theory of life coming from non-living chemicals, otherwise materialism is dead in the water. For evolutionists to believe in chemical evolution, this is not a position they got from science, but a position they got from blind faith. They're basically having to believe in miracles because it's not real chemistry that they can appeal to.
Evolutionists often accuse creationists of believing in a God of the gaps, where they say that we claim God just did it that way when we run into some process that we don't quite understand. But very often when faced with an unanswerable question, a problem like the origin of life, evolutionists invoke their own Darwin of the gaps. See, everything we know about the laws of chemistry, about genetics and statistics and information theory argues against any life from non-life idea. But an evolutionist must believe that scientific laws are violated for life to arise from non-living chemicals. That sounds like faith to me.
I think one of the biggest challenges for evolution is the origin of the first life. Life supposedly coming forth from a chemical soup. Scientists have had decades to reproduce this in a laboratory, but they haven't been able to do that, despite all the time they've had, despite all the great equipment. And I think that's tremendous evidence that life can't evolve from a chemical soup.
Many evolutionists admit that life from non-living chemicals is a huge problem for life on Earth. So they've resorted to the idea that maybe life came from outside the Earth, and this is called panspermia. Now, there are two versions of this one. One is undirected, which just says that chemical evolution happened somewhere and life was naturally seeded onto Earth somehow. Now, this doesn't actually solve the problem of life from non-living chemicals. It just puts it into the unknown. In fact, it puts it beyond science. So you might say it's a Darwin of the gaps theory, not science at all. Another idea is called directed panspermia, where you have intelligent aliens seeding life from outer space onto Earth.
Francis Crick, the primary discoverer of DNA, is one of those scientists who realized it can't happen spontaneously. So he, like other scientists, are imagining that intelligent life from outer space brought life to Earth. And they find that more credible than the idea of spontaneous origin of life.
So two thoughts here. The little cartoon, though, did you catch the little cartoon? I think it summed up the whole argument quite well. If you can see, the scientists, look at the apparatus over here. This is 60 years worth of study. This is 60 years worth of research. This is committed to proving that life can come from non-living chemicals. And the government's paying for that? If I can just synthesize life here, then I'll have proven that no intelligence was necessary to form life in the beginning. But let's be honest. In perfect conditions, precisely controlling every possible factor, we have been unable to replicate, in spite of over 60 years of trying, what supposedly happened in natural, imperfect conditions. I have a theory as to why we cannot replicate what happened in the wild in the lab, because it's not what happened. The second thought. Scientists recognizing the information issue and the necessity for information to come to intellect, many have pushed the problem back to another planet. That does not solve the problem. That just moves the problem to that planet. And we can keep pushing the problem back planet to planet, but sooner or later we've got to define where did the information come from. There was another theory. This was put forth by Dr. Dean Kenyon. It's called chemical predestination. He outlined it in his textbook by the same. He theorized that these amino acids, when brought together, would naturally form themselves, roll themselves into a correct molecule. They were predestined to do it. You just had to get them all together. That was the leading textbook from the 50s and the 60s as to the origin of life and the origin of complex molecules. Over time, though, Kenyon began to reject his own theory because all of his research pointed to the reality that it is not simply how it works. In this next video clip, he's going to talk to you about really what the key issue was that moved him away from this position and caused him to reject his own theory and to go back and say, that textbook that I wrote that's being used in universities all over the country is wrong. And by the way, just keep in mind, I gave you a little estimate here. As you think about the human cell, as you think about a cell, the current estimate is there are about 37.2 trillion in your body. So as you look at the cell tonight, sort of picture that.
If one could get at the origin of the messages, the encoded messages within the living machinery, then you would really be on to something far more intellectually satisfying than this chemical evolution theory.
Yet Kenyon realized that he faced a narrowing set of options. By the 1970s, most researchers had rejected the idea that the information necessary to build the first cell originated by chance alone. To understand why, consider the difficulty of generating just 2 lines of Shakespeare's play Hamlet by dropping Scrabble letters onto a tabletop. Then consider that the specific genetic instructions required to build the proteins in even the simplest one-celled organism would fill hundreds of pages of printed text.
Of course, serious origin of life biologists did not believe that life had arisen by chance alone. Instead, they envisioned natural selection acting on random variations among chemicals to produce the first life. But there was a problem with this proposal.
By definition, natural selection could not have functioned before the existence of the first living cell. For it can only act upon organisms capable of replicating themselves, cells equipped with DNA that pass on their genetic changes to future generations.
Without DNA, there is no self-replication. But without self-replication, there is no natural selection. So you can't use natural selection to explain the origin of DNA without assuming the existence of the very thing you're trying to explain.
Chance, natural selection, and his own theory of self-organization had all failed to explain the origin of genetic information. Now Kenyon saw only one alternative.
We have not the slightest chance of a chemical evolutionary origin for even the simplest of cells. So the concept of the intelligent design of life was immensely attractive to me and made a great deal of sense, as it very closely matched the multiple discoveries in molecular biology.
In the years since Kenyon's rejection of chemical evolution, science has revealed the details of an entire system of information processing that bears the hallmarks of intelligent design. With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
This is absolutely mind-boggling to perceive at this scale of size such a finely tuned apparatus, a device that bears the marks of intelligent design and manufacture. And we have the details of an immensely complex molecular realm of genetic information processing. And it's exactly this new realm of molecular genetics where we see the most compelling evidence of design on the earth.
37.2 trillion of those in your body, little manufacturing facilities. Dealing honest with the facts, there's no naturalistic explanation on how to get the correct amino acids, proteins, RNA, DNA, cell membranes, etc. There's no naturalistic explanation to explain how information came about through random chance and process. The conclusion is obvious. Abiogenesis is, pardon the pun, dead in the water, or dead in the primordial soup if you prefer. So I ask the question, is the theory of abiogenesis scientific? I'll leave it to you. The failure of chemical evolution, though, does not prove the God of the Bible. But at this point, intelligent design is really the only reasonable explanation. And when you think back to our first lesson where we proved the Bible was indeed the inspired Word of God, I think we have all the reason we need to believe the biblical account. With that said, let me give you real quick 3 points from God's version of the story. Ephesians 3:8-9, Paul's writing to the church in Ephesus. He says this, "To me who am less than the least of all the saints, this grace was given that I should preach among the Gentiles the unsearchable riches of Christ, and to make all see what is the fellowship of the mystery, which is from the beginning of the ages, or which from the beginning of ages has been hidden in God, who created all things through Christ." That's point one. From the simplest cell to the most complex animal on this planet, the most complex being on this planet, they were all created by God through Jesus Christ. That's point one. Genesis 1:27, so God created man in His own image. In the image of God, He created him. Male and female, He created them. That's point two. Man, unlike all the rest of the creation, is created in the very image of God. And then finally, my third point, and we'll move along. David writing, Psalm 139, he says this, "For you formed my inward parts, you covered me in my mother's womb. I will praise you, for I am fearfully and wonderfully made. Marvelous are your works, and that my soul knows very well." Pastor talked about this past Sunday. We are indeed fearfully and wonderfully made. We are not the result of random chance and processes. Instead, fearfully and wonderfully made. I'll lead you with just a short video, and we'll end here tonight.
Science ought to be a search for the truth about the world. Now, we shouldn't prejudge what might be true. We shouldn't say, I don't like that explanation, so I'm going to put it to one side. Rather, when we come to a puzzle in nature, we ought to bring to that puzzle every possible cause that might explain it. One of the problems I have with evolutionary theory is it artificially rules out a kind of cause even before the evidence has a chance to speak. And the cause that's ruled out is intelligence.
Since the late 19th century, since the time of Darwin, in fact, in part because of Darwin's writing in The Origin of Species, scientists came to accept a convention, a definition of science that excluded the possibility of design as a scientific explanation. And that convention has a name. It's called methodological naturalism. And it just means that if you're going to be scientific, you must limit yourself to explanations that invoke only natural causes. You can't invoke intelligence as a cause. And yet, curiously, we make inferences to intelligence all the time. It's part of our ordinary reasoning to recognize the effects of intelligence.
Consider, for example, these hieroglyphic messages carved upon the ruins of Egyptian monuments. No one would attribute the shapes and arrangements of these symbols to natural causes like sandstorms or erosion. Instead, we recognize them as the work of ancient scribes, intelligent human agents. Similar reasoning leads us to conclude that the mysterious stone figures on the shores of Easter Island were not formed by the actions of wind and water over great periods of time. Nor do we presume that plants could grow into these familiar shapes without some manner of intelligent guidance.
Of course, we make these inferences all the time, and we know they're correct.
So I'll leave you with this thought. As you look at the human cell, as you look at any cell, you see complexity multiplied by improbability, multiplied by the information contained therein. And there's only one explanation, and that's design. Thank you.